introduce our first speaker as she set up her slides. So Emma Littler, she is a master's student in cognitive neuroscience at Queen's University. She studied fear and anxiety responses with a particular interest on what might cause increased susceptibility or increased resilience to experiencing fear and anxiety. So Emma, I'm looking forward to your talk.
Thank you so much. So I'll just share my screen. All right, can everyone see that?
Yeah.
Okay, so thank you so much for that great introduction. So yeah, like I said, I'll be talking about the influence of individually ventilated cage conditions on rats' anxiety-related behaviors. Before we jump into some of the background, I just want to give you guys a brief overview of sort of why this research is important. So all research in Canada is overseen by the Canadian Council on Animal Care, otherwise known as the CCAC, and they operate with the principles of replacement, refinement, and reduction. And today we're going to be focusing on the topic of refinement. So the CCAC defines refinement as the modification of husbandry or experimental procedures to minimize pain and distress in research animals. And as part of the goal of refinement, research equipment is constantly evolving to better protect research animals. And one major component of this is the refinement to rodent housing equipment. However, it is also very important to note that rodent behavior can be altered by the type of housing, and that's what we'll be talking about today. So one somewhat recent refinement to rodent housing equipment is the development of individually ventilated cages, or IVCs. And in many years, lots of research facilities are adopting the use of these IVCs over conventional open top cages. As you can see in the picture at the top with an open top cage, they're a shoebox style cage featuring a wire lid that permits the free transfer of air from inside the cage to outside the cage, whereas the IVC, shown by the picture on the bottom, is hermetically sealed and is thus under forced air ventilation. The use of IVCs does have very clear benefits. The forced air ventilation and the hermetic seal decreases the spread of pathogens between the animals. It allows for very efficient removal of CO2 buildup inside the cage and allows for very precise temperature and humidity control. However, there have also been some unanticipated drawbacks to the use of IVCs. So there is some evidence to suggest that rodents housed in IVCs behave differently than rodents that are housed in conventional open top cages. For example, Lodge and colleagues found that when they switched over from the use of conventional cages to IVCs, the behavioral phenotype of their mouse model of schizophrenia vanished. Similarly, when Shan et al. Switched to IVCs, the behavioral phenotype of their serotonin transporter knockout rats was also eliminated. And both researchers suggested that IVCs may be increasing rodent stress levels and thus disrupting typical behavior, but the exact cause for this increase remains unknown. So one cage condition that I mentioned that is unique to IVCs is this forced air ventilation system. And this has been posed as a possible explanation for the increased stress levels. The forced air system introduces variables such as draughts, noises, and vibrations that were never present in the conventional open top cages. So the forced air ventilation used for IVCs can either be operated under two different pressure settings, negative pressure and positive pressure. Under positive pressure, air is pushed into the cage through the ventilation system and then passively diffuses out of the cage through a filtered lid at the top. And we call this positive pressure because the pressure inside the cage is higher than the pressure outside the cage. It has been shown to be effective in reducing the spread of diseases and infections between the animals, since they're receiving a constant stream of fresh filtered air and they're never actually exposed to the air of other animals around them. However, in positive pressure, there's a high number of air changes per hour inside the cage and increased drafts, which can lead to dried out bedding, which has been linked to increased aggressive behavior. And additionally, there is some research to show that rodents actually prefer cages with a fewer amount of air changes per hour. So in contrast to positive pressure, under negative pressure, air is being sucked out of the cage through the ventilation system, but passively diffuses in through the filtered lid at the top. And again, we call this negative pressure because the pressure inside the cage is negative of that of the pressure outside the cage. This has been shown to be reducing the exposure to allergens for those of us who work with the animals, since all the air from inside the cage is being sucked into the ventilation system. So as the environments of these two cages is quite different, one of the first goals of this study was to examine if the anxiety-related behavior of rats differs when they're housed in IVCs under positive or negative pressure. And like I said, since some research has suggested that rodents prefer environments with a fewer amount of air changes per hour and fewer drafts and vibrations, we predicted that animals housed under positive pressure might exhibit higher anxiety-like behavior. So as researchers who are ultimately interested in studying stress and anxiety, we're also interested in looking at any other housing conditions that could possibly impact anxiety-like behavior. And one variable of particular interest to our lab is the social housing of rodents. Social housing shows a very robust stress protective effect on many social species, including rodents, and is a phenomenon termed social buffering. So rodents who are pair housed usually show decreased anxiety-like behavior compared to rodents that are single housed. And this effect is very well documented in traditional open top cages. However, at present, it's not known if this social buffering effect carries over to rodents that are housed in IVCs. And so an additional goal of this study was to examine any social buffering effects in rodents housed in IVCs. So while many biological pathways likely contribute to the phenomenon of social buffering, a large amount of evidence suggests that the oxytocin system plays a major role. And oxytocin is a neuropeptide that is primarily produced in the paraventricular nucleus and the supraoptic nucleus of the hypothalamus. It's released in response to stressful stimuli and acts as an anxiolytic, thus reducing anxiety. It's also implicated in prosocial behaviors and is released in response to tactile stimulation. And this is important because when rats are pair housed, they engage in physical play behaviors and thus tactile stimulation that releases oxytocin. And additionally, when more oxytocin is being released, more is actually being produced. And so we would expect that pair housed animals would have a higher number of oxytocin producing neurons due to upregulation of the oxytocin system. And so since we're ultimately concerned with understanding what IVC housing conditions might be contributing to stress, and if the social housing of rodents can reduce stress levels in IVCs, the final goal of this study was to examine if the oxytocin system is implicated in any of these variables. So to summarize, our first goal was to examine the influence of air pressure on behavior. As I mentioned, positive pressure environments are possibly more stressful. And so we predicted that rats housed under positive pressure would exhibit higher anxiety-like behavior. A second goal was to examine any effects of social buffering on anxiety-like behavior. And since social buffering is very well documented in open top cages, we also predicted that rats pair housed in IVCs would also exhibit lower anxiety-like behavior. And finally, our last goal was to explore the influence of the oxytocin system on behavior. And we predicted that pair-housed rats would have a higher number of oxytocin producing neurons in the PVN due to upregulation of the oxytocin system. So 40 male Long Evans rats were used for this study, and upon arrival to our animal facility, they were randomly assigned to their housing conditions in IVCs. Half were assigned to housing under negative pressure and half were assigned to housing under positive pressure. And then within that, half were assigned to pair housing and half were assigned to single housing, and that resulted in the following groups and ends. So after remaining in their housing conditions for roughly 5 weeks, all animals underwent behavioral testing in the elevated plus maze, which I'll walk you through on the next slide. And then after behavioral testing, their brains were harvested and underwent immunohistochemical staining for oxytocin producing neurons in the paraventricular nucleus. So the elevated plus maze is a wooden maze that contains 2 closed arms and 2 open arms in the shape of a cross, like you can see in the picture. And during testing, rats are placed in the center of the maze and they're left to explore for about 5 minutes. And this test relies on rodents' innate fear of open spaces and their tendency to spend more time in dark and enclosed spaces where they're theoretically more protected from potential predation. When placed in the maze, rats tend to spend the majority of the 5 minutes in the closed arms. And so any level of open arm activity can then be used as a measure of anxiety-like behavior, with more open arm activity indicating less anxiety-like behavior. And the most common measure that we use to quantify this is the percentage of time that they spend in the open arms. So the results from the EPM didn't actually reveal any main effects of housing or air pressure on their own, but did reveal a significant interaction. And the interaction was followed up with post hoc tests that revealed that only under positive pressure was there a main effect of housing, such that pair housed rats spent more time on the open arms than single housed rats under positive pressure. And these results are actually quite interesting because we did originally predict that there would be an overall effect of housing with all pair housed animals spending more time on the open arms, but we only see this effect under positive pressure. And this leads us to believe, like we originally predicted, that positive pressure is a more stressful environment. And as you can see in the graph, under positive pressure, the single-housed rat spent the least amount of time on the open arm. And that maybe due to this high stressful environment, the effects of the social buffering are more pronounced. So I'll go over the results from the oxytocin. So contrary to what we originally thought, there were actually no differences in the overall number of oxytocin producing neurons between groups. We originally did think that pair-housed rats would have a higher number due to upregulation of the oxytocin system. However, we did see a significant positive correlation between the number of oxytocin producing neurons and open arm activity in the EPM, with more neurons being correlated with more open arm activity. And this further suggests that oxytocin has stress reducing properties. So to summarize, it does seem like positive pressure is a more stressful environment for rodents. As I said earlier, drafts inside the cages and the higher number of air changes per hour may be responsible for this. And this is in line with previous evidence that actually shows that mice living in IVCs with a higher number of air changes per hour displayed increased anxiety-like behavior on the EPM. We only saw effects of social buffering under positive pressure. So if positive pressure is truly a more stressful environment, it may be more sensitive to the stress reducing effects of social buffering. However, it is still odd that we didn't see an overall effect of pair housing because it is a very well documented phenomenon. And again, this does lead us to believe that IVCs might be influencing typical rodent behavior in general. And finally, although we didn't see any differences in the number of oxytocin producing neurons between groups, there was a positive correlation between the number of neurons and open arm activity. And this is in line with previous evidence that also shows similar findings. So before I finish, I just want to leave you with some thought-provoking questions and some future directions. So first, what are the implications of these findings for the well-being of laboratory rodents? The ultimate goal is the refinement of housing conditions to better serve laboratory animals and reduce any suffering. So future research should explore how housing conditions can impact rodent stress levels. And our lab actually has future plans to compare the use of IVCs to open top cages. And second, how does this impact our ability to conduct behavioral research? Because it does seem like IVCs are impacting typical behavior. And as behavioral researchers, anything that's impacting typical rodent behavior can confound our results. And so more future research should look into how any environmental factors, including housing conditions, can impact rodent behavior. So thank you for listening. And if you have any questions, let me know.
Thank you very much for the interesting talk. Let's see if we have questions. Please post questions to the Q&A session, and while we're waiting for the questions, I will ask you something. This is very interesting to me, and I think like rodents are relatively more olfactory oriented, but would that, like when they're housed individually, can they see other rats? Would that help?
So that's actually a really interesting question and something that I didn't really touch on in the presentation because I didn't have time. But the thing with IVCs is that because they're hermetically sealed, they can't smell any other rodents around them. And because rodents are such social animals and do rely on a lot of olfactory cues, that is another variable that we're actually looking at, is the transmission of olfactory cues between the animals. When rodents are housed in cages next to each other, they don't have great vision, so they actually can't see any of the animals around them, but they do get olfactory cues if they're housed in those conventional cages where the air can transfer between both of them.
Yeah, I forgot that they can't really see that well.
Yeah.
Yeah, that's really interesting. I think I have other questions if there's nobody else is asking. And do you think the positive pressure that's maybe induced, like matters more, is they also probably need to smell themselves? I don't know. Would that be the case?
Smell themselves? Like how so?
If it is positively ventilated, it would seem that whatever their own odor they're producing, like it will feel like a home cage. It probably actually smells like themselves. This is just
No, that's an interesting question because under both pressure systems, like the air is being removed from the cage like either way. Under positive pressure, it's a little bit more intense because the air is being like pushed in versus like sucked out. I don't really know much about like how rodents sort of like nest in their house or nest in their cages and then like respond to like what they've deemed as like their like safe space. I'm not really sure, but that's actually a really interesting thing to think about, because in open top cages, they would not be getting that like change in air. And so I guess their cage would smell similar to them all the time.
Very cool.
Yeah.
All right, now it's time for us to move on to our